Hello my dear children, just give me a quick second, I'll just reload. Yes, am I audible? Am I visible? Is it good to go? Yes. Hi Elena. Hi Elena. I hope you're good. A very big hello my dear children. So finally, I suppose the major big subjects which are there, they are over. And now you would be having languages, right? So I think there's a problem with my net, or let me see. Okay, just give me a second, okay? If you're able to see me, just give me a second because I'm not able to see your, yes. Sailani is also there. Hi Sailani. I hope children, I'm audible. Hi Rekha R. Okay. So first of all, how was the paper? Was it difficult? Was it easy? How was it? Well, you know what, the good part of the story is that whatever we discussed has come, and that's a very big thing, right? So I'm just waiting for everyone else also to join, for those of you who have just got the message. Okay, I think we'll begin off. Hi ma'am, how are you? Oh, that's so sweet of asking. So sweet of you. Well, I am doing pretty fine now. Much better. I hope you are also doing great. Yes. So, starting off with the first question that we have. This is a conceptually direct question. So, I have taught you over here. So, for each and everything which I have taught, I have brought in a proof to tell you that yes, this came from what we learned together. So, I was pretty happy seeing the question paper. Two questions are there which were more like little bit confusing in MCQs. Rest all of it was super good. Hi Shazam. Hi Rekha. Okay, so going ahead with the first question that we have, the functions of tapetum in microsporangium is to what? Nourish the developing pollen grains, and this is exactly that we learned in our live session. So I had taken two sessions, two lives, part 1, part 2. Okay, one live was there and that was part 1. Another one was recorded and that was part 2, and from all of it I saw the questions. Okay, so going ahead, the first answer would be A, my dear children, to nourish the pollen grain. That is the function of tapetum. It does not help in dehiscence of anther. It does not help in protection and dehiscence. It is not for dehiscence as well as nourishment. Dehiscence is basically for the other three things, epidermis, endothecium as well as midterm. Renuka, please ask me the doubt that you're having. Going ahead with a question, this is a direct question, we solved it. This is question number 2 while we dealt with human reproduction. The question says the cells that secrete testosterone hormones are, it is what? None other than Leydig cells. Leydig cells are the one that releases testosterone hormone, or basically the male sex hormones. Okay? Renuka, you can go ahead and ask the doubt. And silently, I am super good. I hope you're also great. Alina says, paper was easy, but 5 marker was interesting yet tricky. Yes, it was a bit, but I hope you guys were able to do it. Okay. Let's go ahead with the next question. This is again related to the concepts that we study, and that is statement number 1 says amniocentesis is legally banned for sex determination. So here there's a statutory ban on amniocentesis. Because of what? Because it was used for determining the sex of the child. So obviously, yes, it is correct. Amniocentesis was misused for aborting normal female fetus. Yes, when the when the sex of the child was being identified, the child was being killed. Okay. Renuka, I'll slow it down, dear. You can just tell me, okay? So I have did correct this MCQ. Great. So we'll calculate our marks, okay? So for 3A is the correct answer. For 3A is the correct answer. I hope you guys are getting it. If you want, I can go behind. For second, B was the correct option. For the first one, A is the correct option. Everything that we have learned. Okay? Going ahead. The next one, the fourth question, which of the, okay, just a second. So I'll mark this off. Okay. Which of the given genotypes is or are the dihybrids? Is or are the dihybrids? Now this is a what do you call tricky question. They're asking you which among them is a dihybrid. Dihybrid means basically you are talking about pair of contrasting traits, but you should also understand that basically if at all we make a plant undergo dihybrid cross, and if we want a ratio like this, okay, if we want a ratio like this, then we have to take capital R, capital Y, small y. The alleles for both the traits should be heterozygous in condition. This would be a proper dihybrid. Okay, this would be a proper dihybrid. Actually saying all of this could have been a correct answer, but over here, according to the PU board, they have given this as the correct answer. Hi Aryan, I hope you're doing great. 5-marker part, we'll come to it, Ramu. Okay, so the answer for this question is C. If at all you have went with D option, all of this, it would be wrong. The answer for this particular question would be C. Why? Because only if it is C, then only in the dihybrid cross experiment, my dear children, we will get a ratio like this. That is the reason. I hope you guys are clear with that. Yes. Okay. Next question over here again. Still I didn't answer 5 marks. Okay. So 5 marks is something where you guys I think are getting tricked. Let's see. We'll come to that, okay? Now over here, the number of nucleosomes required to pack the 4.6 into 10 to the power 6 base pairs of DNA in E. coli is. So my dear children, I explained you, this is again a conceptual direct question. I explained you while I explained about nucleosomes or the structure difference between difference between eukaryotes and prokaryotes packaging. I told that prokaryotes, that is E. coli, they do not have nucleosomes. They do not have histone proteins because they do not have a nucleus. So basically they do not need to arrange everything at small place. So there is no nucleus, there is no compact DNA, nothing like that. So your answer for this question would be D, 0. My lab says I wrote the answer as D. I think that is for the question number 4. Well, it happens, it's totally fine. Listen, it's preparatory examination. It's preparatory examination, chill. You gave your best, and I could clearly see that a lot of you came in the live, attempted the live, you got an understanding of a lot of things. It is going to help you out. Okay? Fine. So the answer for this question is 0. So again, I always tell you, whenever you solve a conceptual question, please understand the concepts well. If you understand the concepts well, you will be able to do questions also very easily. Okay? Going ahead with the next question. This is a direct question that we solved. Okay, so I gave you the PDF also for this. Single step large mutation leading to speciation. A single step large mutation which leads to speciation, formation of new species, will be what? Saltation. It will be saltation. It was the 15th question of evolution that we solved. Got it? Okay, so now since it is going very, momentarily, I want you guys to give me a thumbs up after every question. So this question is done. Can I move to 7th question? If it's fine. Shall I go ahead with the 7th question? This is very easy. Yes, Sailani. This is pretty much easy, very much easy. I hope I can go with the 7th question. Again, this question is a direct question. We solved it. This was a 9th question of the chapter Human Health and Diseases. So which of the following is an autoimmune disease? Autoimmune disease means basically where your cells, your body cells are not able to recognize what is self, what is non-self, and ends up in killing our own body cells. So the answer for this question is rheumatoid arthritis. We discussed this. This was a 9th question in our live session. Yes, my lamp, Alana, thank you so much here for telling me that you're understanding, and we can go ahead, right? So question number 8. Okay, question number 8. Injection of antitoxin in tetanus confers which type of immunization? Again, this also we just did it. Okay, we just did it. So over here there is one catch to the story. They have changed it slightly, and that is injection of antitoxins is basically you're giving ready-made antibodies. So obviously it has to be passive immunity. But there are two types of passive things given over here. One is natural passive and the other one is artificial passive. One is natural, one is artificial. Natural means basically naturally you're giving ready-made antibodies, for example. So the milk you are giving from mother to the child, that is natural passive immunization. Artificial means basically you are giving from outside, somewhere outside. So over here in antitoxin we are receiving it from outside, which is why it comes out to be artificial passive immunity or passive immunization. Got it? Did you get it? All cool? Give me a thumbs up. If it is cool, give me a thumbs up. So we did the exact question. They didn't change anything. They changed the options a bit. Okay, they made it a bit more tricky so that you get an understanding that in biology for boards we should not take this subject lightly, right? I was confused between B and D, but thank God I remember this question from MQQ. Oh, that's so nice. But ma'am, okay. Amreen, dear, I do not know which question are you solving as of now. My lamp, Alana, yes. I hope you got it. Going ahead to the next question. Question number 9. Again, we discussed it while we solved about the question about penicillin. This was the recorded video. So over here, Penicillium notatum is what? Penicillium notatum is a fungi. Fungi basically a mold, right? Fungi means basically it is a mold. So the answer for this question would be what? Mold. And what does it release? It releases penicillin, which is the first antibiotic, that we are having. Okay, cool. So is there anyone who says that biology examination was easy, or do you think it's still difficult, very much difficult for you guys? 7th question, it is rheumatoid arthritis, dear Likhit. Okay. Now this question, again a direct question from the live, we discussed it, and that is which of the following is the correct order of processing of PCR? So it is what? Denaturation, then annealing, then extension. First of all you separate the DNA, then you add the primers, and that is known as annealing. Later on you extend it. Okay, so over here this is your answer. This is a direct question. They just have changed the options. This is from the question bank, and yes, Sileni, yeah. The MCQs are some MCQs are literally very easy. Yes, actually, to be honest, I feel the whole paper was not too difficult. It's just that if our concepts are clear, we will be able to answer it. Okay, that's the only thing that I could find out. Okay, my lamb says 5-marker session was quite tricky, then rest all were easy. Yes, that's the spirit. Okay, great. Now question number 11. Now this is a question where I really hope you're not losing your marks. So which of the following facilitates the even mixing and oxygen availability throughout the bioreactor? So over here we have studied that impeller is something that mixes everything. Now when we go to NCERT and read the NCERT book, they put down the term, the stirrer facilitates even mixing and oxygen availability throughout the bioreactor. So stirrer is actually the other name for impeller itself, but in your NCERT, instead of mentioning about, in the diagram they have given impeller, but while they have written, okay, they mentioned the stirrer. So which is why the answer for this question would not be impeller but will be stirrer. So this is something that I want to tell you. Alana, I'll get 65 plus marks. I will only lose only in 5 marks, maybe 2 or 3 marks. I hope you are not going to lose that also. I genuinely wish you get all the marks. Okay? So your answer for this question, 11th question, is B. Am I clear? Can you give me a thumbs up, everyone? Everyone out there, can you give me a thumbs up for this, if it's clear? NCERT is the only way to solve this MCQs. So yes, that is the only way. Knowing the concepts actually is the only way. to score really good in biology. That is why, you know, in biology you cannot go with short videos and explain this and that's over. We cannot do that. We have to take long lives, we have to take long recorded video, and not just about taking it, you guys have to watch it. Okay, don't think that there's a shortcut to getting marks in biology. There is no shortcut. That is why it becomes a big, and sometimes it can be boring as well. But I try my maximum to not make it boring. So Rach Thomas says, Ma'am, Mallu here. Hi. Another Mallu right over here. Okay. Now question number 12, this is a direct question. We discussed it several times. An explant used in the production of virus-free plants in plant tissue culture is. So if at all you are going to take from the plant, some part of the plant, and you have to make sure that you're using in tissue culture and it does not produce virus affected plants, then what part will you take? You always take either apical or axillary meristem. Either from the apex or from the places the branch comes off. These are known as this is apical meristem, this is axillary meristem. So over here you do not have apical meristem as an option, so axillary meristem. So the good part of the story is that these are all direct questions that we have discussed. I hope the next time any exam comes over, you are gonna come and join the live and just do all the questions. Okay, very, very important. And see the recorded videos as well. Okay, so Gagan is over here. Okay, all cool. So 13th question, which of the following graphs illustrates the population growth curve of a single bacterium growing in a flask of ideal medium at optimum temperature over a period of 24 hours. So you need to understand, a single bacteria is growing in an ideal medium. That means it has everything that it wants to grow. That too at an optimum temperature. Again, a very nice temperature where it can grow easily for the period of 24 hours. Where everything is suitable. When everything is suitable, then obviously it is going to grow exponentially. It is going to grow exponentially. There will be a J-shaped curve. And a similar, I have written conceptual direct. As in, I spoke about this concept. I explained you right over here when I solved this question, question number 4. Okay, this is basically here the conditions were that the culture was soon going to end up, which is why what type of growth will you see? If the culture is soon going to end up, then obviously it will be sigmoid, that is logistic growth. Then I explained you that if at all it is not ending up, it is going to show what? Exponential curve. So that is the exact thing that is happening. So your answer for this question would be D. Okay, it would be D. Alaina, ma'am, I would like to thank you because you helped me motivated. I don't know if you remember, you had told that your parents told you that don't think about future, do what you have now, which motivated me. I was scared too. Oh, that's so sweet. Wishing you the best. I hope everything good reaches all of you guys. So my dear children, your answer for this question would be D. Going ahead with the next option. NPP is the result of. NPP, this is conceptual direct. Here while I explain that, I told you NPP is a result of what? GPP, gross primary productivity minus respiratory losses. This is a very direct question, very easy question. So your answer for this question would be C. Your answer for this question becomes C. Okay. Hi Vittal. Vittal is giving me the answers, but I'm not understanding which question's answer. So can you tell me 15, 14, 13? It will be a lot more better. Okay, now the following are the in situ conservation approaches, except again same question, we solved it together. It's a direct question. So the answer for this question is that botanical gardens are not in situ conservation, it is ex situ form of conservation. Ex situ form of conservation. Okay, so your answer for this question is C. Fine, children, understanding? 15 MCQs done. I hope you have scored well. And Alena, I really want to thank you because even now, so that is exactly, you remembered it, that means a lot, and I am really glad all these things that I'm saying, it is there in your head and you guys are being helped with the help of that. Because I remember my father told me when I was going to appear for my boards, for my class 10 boards. So yes. Yes. Now the next is the fill in the blanks question. Most of it again are direct questions. The process of delivery of fetus is known as parturition. During Mesozoic era, Earth was predominated by conifer plant groups. So over here you just have conifers. So please go with conifers. Yes. And white odorless bitter crystalline compound that act as a depressant. We learned depressant. It was heroin. I also told you it was known as smack. So here is your answer. It is smack. Okay, now the genus of the fungi that makes symbiotic association with the roots of higher plants, we all know the answer for this question would be what? It will be glomus. It would be glomus. Okay, and the region of South America that has the greatest biodiversity on earth is Amazon. You do not have any other option that matches them. Okay, so over here your answer for this question would be what? These. So please make sure you go through it. And yes, ma'am, I got 13 correct. Awesome. Awesome, Ayan. And Alina says 20. Whoa. Awesome, awesome, awesome. Okay. My lamb says, oh my God, okay, thank God, 5 on 5 on, what do you call, fill in the blanks. Yes, exactly. That's so great, so great. Rohi says, ma'am, 14 MCQs are correct. You know what? You guys are super. Okay. So here, Vittal, my dear child, if at all in ideal medium they had mentioned that there is limiting resource and things like that, you could have went with sigmoid, but over here they had not mentioned any limiting resources, which is why the answer for that question would be exponential curve. Okay? Shivanand, your voice is really nice, ma'am. Thank you so much, dear. And guys, have you subscribed to the channel? And also, if at all you have not, please do that, because I am pretty sure that the live lecture and the videos, everything has helped you. When I put direct over here, I put direct because I know that I taught it. And as I said, that I have put the proof over here. Anyone who has a feeling that, okay, this was not covered, should understand that this was covered. Probably you might have missed it. Okay, so distinguish between albuminous and non-albuminous seeds. We covered it. It was a 2-marker question. Albuminous seeds means some of the endosperm still leaves behind. Non-albuminous means endosperm is completely used up. Over here, they would have endosperm as a stored medium to provide nutrition. Here, they might have cotyledon or the endosperm. Then give the example. That would be more than enough. This question was from the question bank. Okay, this question was from the question bank. Cool. And my dear children, do not forget to like this video and share it with your friends also who has given biology examination. Let them also know how it is going. You know what? Do not be sad if at all you get your answers wrong. I'm telling you again why. Because this is just a preparatory examination. If you know where you have went wrong, you might be able to correct yourself, right? So list one contraceptive method, each in males and females, with poor reversibility. Reversibility is poor, and this is a contraceptive methods. Ma'am, for albuminous seeds, can coconut be considered as example? Albuminous, yes. Coconut also can be considered as an example. It's a monocot, right? So yes. Okay. So surgical methods, tubectomy and vasectomy. We're cutting the tube, fallopian tube, or we're cutting the vas deferens. Basically, they are going to show what? Less reversibility. So over here, just a second. Yeah. Yes. Here it is clearly written, highly effective, but the reversibility is very poor. Okay, so my dear children, please, I hope you might have written the answer as surgical methods only. You just had to write one. So either what do you call, emergent eye mismatch tubectomy to male. Is it? Oh, it's okay. It's okay. We learn from mistakes, right? But you knew the concept. It's okay. It's okay, Vittal. Fine. Next question. This is conceptual direct question. Something like this we had solved. Write the pedigree symbols for sex unspecified. Sex is unspecified. Basically, that means, how do you symbolize that? You show it like this. Sex is unspecified. Now mating. Mating, how do you show? You just make a square, a round, and this is what? Mating. This is one of the easiest question out there. Okay, this is the easiest question. Writing only the name of the surgical method is fine, right? This is a 2-marker question, so obviously you would have to mention a bit more about the contraceptive method. Okay, simple, beauty. I hope that works. Yes? Now, name any 4 non-human model organisms in which genome is sequenced. Now, this is a new thing that we had not covered, and I have put that over there. So, 4 non-human model organisms in which genome was sequenced in the Human Genome Project were bacteria. So, I'll just show it. It is bacteria, it is yeast, it is Cenorhabditis elegans, okay, and also Drosophila. These are the 4 organisms, and also the plants which are rice and Arabidopsis These are the 4 things that you would have to write. Now, one very important thing, I was quite relaxed when I saw this question, because in your 2-marker session, I believe you just had to write 5 questions. So even if you do not write this but attempt something else, it is totally fine, because we had discussed everything else. So I was kind of happy with that. Okay, I think today I'll sleep peacefully after sacrificing 5 hours of sleep every day. Oh my God, Lamb, please, please sleep. And also, Alayana, you guys sleep and please take care of your health. That's very, very important. Now, what are homologous organs? Please give example of it. So that we discussed over here, bougainvillea and tendril of cucurbita. Both of them are homologous organs. I've explained over here why it is homologous organs, because my dear children, both of them have the basic structure. Okay, they originate from the basic structure but perform different functions later on because they are showing divergent evolution. So this is exactly what you have to write. Make sure you write this and you might have got your marks as well. Now, what are flocks? Mention its significance. Again, a question that we discussed. This came in your MQP2 also. So make sure you write I hope so you have written the same answer. What are flock? Flock is just basically loose aggregates of the aerobic bacteria as well as the fungal filaments coming together. And what are their significance? Mention their significance. They break down organic matter and they reduce the BOD. Lower BOD means what? Less pollution. Higher BOD means high pollution. So all of that. Okay. Cool. Let's go ahead. Now, over here, what exactly is stability for biological community? Even though we did not directly discuss it, we discussed David Tillman, which is why I read conceptual direct. So stability basically means your productivity should not have much variation. It must be either resistant or resilient to occasional disturbance, such as natural or man-made, and also be resistant to what? Invasive species, alien species invasion as well. Okay, Sanskar, when was second preparatory exam? When is second preparatory examination is what you're asking? I hope so. It is soon going to start from 19th of January in most of these schools, PU colleges out there. When are your second preparatory examinations going to start, my dear children? Is there some other date for some people told that it's not happening in the month of Jan for some schools, it is happening in the month of Feb. So please let us know that as well. We will plan our lectures accordingly and could help you out. Okay? Now, the next question are 3-marker questions. So my dear children, I believe if at all in this 2-marker, this question 1, 2, 3, 4 and 5, if you have done this correct, then it's all fine. You do not be worried. You need not be worried about biological community, nor you need to worry about this particular question out there. Okay? Now the next question that we have, just a second, is a 3-marker question. Draw a label diagram of mature embryo sac and label 3 parts. So we know how to draw a mature embryo sac and label its part. We had discussed it. I hope you had done it. Okay, my lamb says ours college is not going to conduct second preparatory. Yes, so in some colleges that is not even happening. So yes. Next one, mention the significant embryonic development at the end of 1st, 3rd and 6th month of pregnancy. Again, we discussed it over here. So first, after the end of 1st month, you have heart bean formation. After the end of 3rd month, major organs, limbs, external genitalia, everything is being formed. And after the end of 6th month, you would be having what? Fine hair covering your body, eyelids are going to be separated and eyelashes are going to be formed. Okay? Vittal, that day morning, ma'am, you have taken live without sound. Vittal, actually what happened is that I recorded it. Okay, that was a recorded video, not a live. And so there was a problem while rendering the video. There was some processing errors that happened. So while I recorded it, there was sound. But when they uploaded it, somehow there was some technical glitches out there, which is why the voice went away. And I was literally very sad. In the morning, 4:30, I got the message that Jasmine, there is no sound in that video. So I was kind of very sad. Okay. Ma'am, 13th question, KVUS is smart. I'll get back to it. When will be practicals, ma'am? So it depends upon college to college, but what I get to understand is from the month of Feb, you have your practicals. But please let us know if at all there is some other date when you have it. Okay? Now, the next question that we have, so basically, out of 3-marker, 1 question would be easily done. Second question can also be easily done. We discussed it. The third question, list 3 principles through which one could be free of STIs. Again, we discussed it. Okay, this was in the sheet. So, avoiding multiple partners, correct and consistent use of condoms, and prompt medical checkups and the treatment. These are the few points that you need to write. These are the 3 principles through which one could be free of STIs. Now next thing, 31st, state Hardy-Weinberg principle, mention 4 factors which disturb the genetic equilibrium. Very easy question. What is Hardy-Weinberg principle? It says that the genetic allelic frequency is going to remain from generation to generation. Okay, then there are effects, there are things that affects gene flow, genetic drift, mutations, genetic recombinations, natural selection, all of it are going to be what? The new 4 factors out there which disturbs the genetic equilibrium in a population. Okay, so I hope you have written that as well. Okay, now next, name the diseases caused by the following pathogens. So 4 questions in the 3 marker, we have got it correct already. Now we are going ahead. Haemophilus influenzae. I told you while we discussed this particular thing that Haemophilus influenzae causes what? It is Haemophilus pneumonia given over here, that's why it was wrong. Haemophilus influenzae causes pneumonia, that is the name of the disease. Rhinoviruses causes what? Common cold. And Plasmodium, we all know it causes malaria. It causes malaria. Okay? Got it? Okay. Going ahead, ma'am, should we explain the terms as well in Hardy-Weinberg? Yes, yes, yes. Okay, just a second. You should be explaining that at least a bit. I don't think they might have cut your marks, but in boards, please make sure you at least have a 1-liner explanation of each of them. It's a 3-marker question, no. So while you say Hardy, I do not know how well you explain the Hardy-Weinberg principle. If at all you have explained it well, and then you have mentioned the 4 factors, then it is totally fine. But if there is no much explanation in Hardy-Weinberg, and then you have not explained here also, then I'm a little doubtful. But I hope they would not cut your marks because it's just preparatory examination, or they might because it's a preparatory examination is for preparing you guys. But please know in handy that in your boards, do not be confused, write answers with the time limit in your head, and at least 1 line. That would be very much helpful. Okay? Okay. Going ahead, name the disease caused. Oh, we did it, right? Okay. Mention the microbe that produces the following bioactive molecules. The same question we came. Okay, same question was discussed as well, my dear children. Cyclosporin, streptokinase, basically who produces it? It is Streptococcus. Cyclosporin A is produced by Trichoderma polysporum. Statin is produced by Monascus purpureus. Monascus purpureus. So your answer for that particular question also comes out to there. May last main is applied. Please explain that. Oh, I'm so sorry, I didn't get your question. Can you please come back to it? KVUS smart classes, ma'am, 13th question, please. I'll come back to it. Okay, just give me a second. I will just finish it off and I'll come back. Okay, the whole paper is going to be finished in few minutes. Now going ahead, draw a label diagram of pyramid of number in a terrestrial ecosystem. So if at all you are mentioning grassland ecosystem, then it looks like this. Listen, if at all you are making forest ecosystem, then it will be inverted pyramid. I explained you that. Okay, I explained you that if it is a forest ecosystem, 1 tree will be there, producer will be less, but the number of organisms feeding on the producer would be more. But over here in grassland ecosystem, you can make like this. It's also an example of terrestrial ecosystem. It is also an example of terrestrial ecosystem. So please mention, I hope you have mentioned it, it's a grassland ecosystem, and then put like this, primary producers, and then primary consumer, secondary consumer, tertiary consumer, and put down the numbers. Okay, I hope that's clear. Yes. No. Ma'am, should we take the same numbers? No. You just have to make sure that the number over here is the highest number out there. And then it should be getting lower. Okay, and from here to here, there should usually the apex predators, my dear children, it's not too much. They're very less in number. So keep that in your mind. You're not going to cut your marks out there, but please have that in your head. And please mention grassland ecosystem. Okay, not all the terrestrial ecosystem will be having an upright pyramid. Okay. And if at all you can put down grasses, then whatever organism's name is there, all of it. Fine. I hope that's clear. Going ahead with the next question. Now this is a 5 marker which you thought was tricky. So why the analysis of the 3 markers that we have got? I again understand that there were 5 3 markers that you can easily attempt and get marks. We had discussed it also. 10% law, no, 10% law is there in the pyramid of energy. Here pyramid of energy does not apply. Okay, but just understand that the number in the apex predators, for example, if at all you take in general sense, the number of tigers will be very much less in terms of number of producers, that is trees or anything out there, right? Obviously, or the amount of grass which is there. So apex predators are always less, and the reason behind that is 10% energy, obviously. Okay? Shauna, paper was easy. Yay! Finally, there is someone who says that paper was easy, not tricky and anything out there. But yes, I get your point also, even if you say it's tricky. So 5 marker, the first question that we have is super easy question. Draw a diagrammatic sectional view of the human female reproductive system and label 5 parts. This is the human female reproductive system, and what we have to do? We have to label the parts. Okay, I hope you did that. We did that in 5 marker session. We discussed about that already. Now, represent the schematically the result of gene interaction in snapdragon with respect to flower color. That means you are asked about what? You are asked about incomplete dominance. So again, we have discussed it. This is a direct question. We discussed it in our live session. So my dear children, I hope you had made it. You had made everything that is the first generation out there, or the P generation out there, parental generation, red and white gametes are this, and the F1 generation comes out to be all pink in color. Then we cross it, these are the gametes. F2 generation would be this, 1:2:1 with the phenotypic ratio as well as the genotypic ratio. I hope you guys mentioned all the ratios out there. Okay, so this is how you get the marks for this. So out of the 5 marker, I believe 4 5 marker has to be done, right? 3 from here and 1 from there. So 2 questions are already done of 5 marker. Now, differentiate between Mendelian disorders and chromosomal disorders with an example. So, conceptual clarity was given when this, Rage Thomas says, I was happy when this question came. Easy, yes. Okay, Viju Baba, ma'am, please explain 5 marks answer to question. Okay, the second question, I think in 5 marker, okay, I hope you are asking about part 2 because, yes. Anyways, so Mendelian disorders basically means if at all in a chromosome one gene is being affected, that is Mendelian disorders. Now chromosomal disorders basically means that abnormal number or the structure of chromosomes is there, that's why you get the disorder. Now here in Mendelian it can be dominant or recessive, right? Sex-linked recessive disorder, sex-linked dominant disorder and all of that. But here it occurs due to non-disjunction during what? Meiosis. No proper, no proper division during meiosis. Okay, and now affect specific traits or functions over here in Mendelian disorders, but over here multiple traits are being affected. Why? Because you're talking about a whole chromosome being lost or being added up. That's the reason. And if at all you want to give the examples, then Mendelian disorder example is sickle cell anemia or hemophilia, anything like that. Chromosomal disorders, Down syndrome, Turner syndrome, Klinefelter's, all of it. Okay, Crick X said, oh my god, the name is too difficult to pronounce, says it's easy, but last main was a bit tough. Yes, I get it. Yes, last one a bit tough, but not too tough. If at all, as I said, if your concepts are strong, that void might have been easy for you. Now, B part of this is write the karyotype of the chromosomal disorders with trisomy. Now, chromosomal disorders with trisomy, we discussed it during live. There are 2. One is Down syndrome and one is Klinefelter's. In Down syndrome, we have extra 21st chromosome. There is extra 21st chromosome. So, the number of autosome, 21st chromosomes means 22 chromosomes are autosomes, my dear children. 22 pair of chromosomes are autosome. 23rd pair is the sex chromosome. So, if you have one more extra chromosome, okay, and 22 pair was there. 22 into 2 is basically 44 chromosomes were already there, but 1 is extra, so 45A, that is 45 autosomes plus XY or plus XX. Down syndrome patient who is a male, Down syndrome patient who is a female. That is one thing. Now, one more trisomy is in Klinefelter syndrome. In Klinefelter syndrome, you have one extra addition of X chromosome. So, 44 autosomes normally you have, but over here the sex chromosome, you have an extra X chromosome. You can also write it like this, 47, XXY. In total, if you see, the number of chromosomes are 44, 45, 46, 47. That is why you can write like this also. Okay? Ma'am, then I am losing only 2 marks. Yes. So, TM Darshan, B question karyotype. I hope now karyotype is clear to you, dear. Darshan, as well as I think Viju was also asking the same thing only. Yes. So, the karyotype one, I hope it's clear to you now. If yes, can you give me a thumbs up? Can you give me a thumbs up? Can I move ahead with the next question? Everything was correct till now. And in 6 men I'll lose 2 marks. We'll see if at all we'll lose marks or not. Okay. Now explain the salient features of the double helix structure of the DNA. We can explain this, that polynucleotide chains are there. One is from 5' to 3', another from 3' to 5'. Sugar phosphate backbone is there. There is base pairing between adenine and thymine, cytosine and guanine. Usually one complete turn is 3.4 nanometer long. 10 base pairs are there per turn, and in between each of them you have what? 0.34 nanometers. This is an easy question. Okay, so basically if at all I see the 5 markers, this is where you can score 1, 2, and if at all you can, then 3 over here, and this is the 4th one. You have one more option. This is so easy. I have told you 10,000 times that this is going to come. So schematically represent the replication of HIV virus in a macrophage. This I explained you this as well as this. So I hope this is particularly being done. You have to properly mention viral RNA was there. This RNA was inserted into the cell. It became DNA. How? With the help of reverse transcript enzyme. And now this DNA was incorporated into the nucleus of a host. And then now with the machinery of the host, you got the viral RNA and the viral proteins. Yes? Simple beauty? Yes. Okay. So this is also something that you can get correct. So 4, though easily, my dear children, there are so many options. Now here also BT cotton, again so many times. A direct question, we discussed it so many times. So explain the BT cotton, how it is there. What is BT cotton? Bacillus thuringiensis, we are taking cry gene from them and inserting into the plant, making it resistant to pest. You have to mention that cry 1AC, cry 2AB from thuringiensis is coding for toxin. These toxins are what? Inactive toxin. They become active when an organism has it. In the gut, in the mid-gut region of the organism, of the bacteria, it becomes active and creates spores, and now these spores, my dear children, leads to death. Okay, so explain like that, give some examples of it, and it's all done. Okay, were you able to do it? So until now I don't see any difficulty over here. Okay, chalo. Now, parasitic mode of life ensures free lodging and meals. Justify the statement by listing the special adaptations developed by parasites. Again, we discussed it. Okay, there are certain features. List any 3 parasitic adaptation: loss of unnecessary sense organs, presence of adhesive suckers, things like that, a high reproductive capacity, complex life cycle including intermediate host, and also, my dear children, yeah, that's all. So if at all you have written those points, you get the marks. 41, I wrote about modification of parasite hooks and suckers, digestive system not well developed. Yeah, that would be more than enough. Yes, that would be more than enough. Gagan, sure, I'll get 75 plus. Abba, the paper was out of 70, da. Okay, I'm so happy that you are this much confident that you are getting more than full on full. Okay, for final it will be easy like this question paper, ma'am. So, listen, so some people have that thing that, okay, it was difficult. Some people say it is tricky. So expect it to be a mix of both. It might be tricky because in biology, the only way you can mess up with children is that make the question a bit more trickier, right? So they might do it, be prepared for it, okay? But I'm pretty sure you guys will ace it. So previous year, I remember a lot of you scoring 100 on 100. And this year, I want to see the same. I really want to see the same. Okay, now this is a 5-marker question. Again, the next part, the 5-marker question, where you have to attempt any one question, right? And I suppose this is one where you think it's, what do you call, a bit difficult. Zygote is 5N and PEN is 7N. Exactly. So question says, double fertilization is a unique feature of angiosperms, and the products of this double fertilization are zygote and primary endosperm nucleus. In the context of this, when a hexaploid plant, hexaploid means basically 6N is the ploidy. Hexaploid plant pollinates the tetraploid plant. Tetra means basically it is 4N. Find out the ploidy of the zygote and the primary endosperm cell. Listen, if 6N, suppose your male plant, okay, suppose your male plant is having 6N, it will undergo meiosis, and it will have half the number of chromosomes in the male gamete. This is what the male plant, it has to make the gametes, and gametes always have half the number, so it is 3N. Your male gamete is having triploid nature, 3N. Now female is tetraploid, it is 4N. It undergoes meiosis, the egg will be having half of it, so it will be 2N. It will be 2N, right? Now the male gamete and the female gamete, when it comes together, 3 and 2, 3 plus 2, not into 2, 3 plus 2, it will be what? it will be 5n. Your zygote, the male and female gamete, when it comes together, you get the zygote, it will be 5n in nature. Now, if you're talking about the primary endosperm nucleus, how the primary endosperm nucleus form? When the fusion of one male gamete, another male gamete happens with the polar nuclei. Remember, one polar nuclei is having diploid, another polar nuclei which is also there, it is also having diploid nature. Okay, all the cells are diploid in nature, right? Nucleus at least are diploid in nature. Now you add one more male gamete over here. Male gamete was 3n. So 3 plus 2 plus 2 is equals to what? 7. 3 plus 2 plus 2 is equals to what? 7. So that is why 3 plus 2 plus 2. This is triple fusion. These two are polar nuclei and this is the male gamete. And that is why you get primary endosperm nucleus with 7n. Got it? Yes, 7n polar nucleus Shaunak and 5 zygote. Exactly. Vital zygote, Vital and Shaunak, everyone is correct. Gagan, 65 plus plus, my bad, I thought it was 90. It's totally fine. Children, I'm reminding you again, did you share this live lecture with your friends also who are in need? Please share with them and ask them to not be sad about anything, do not be worried or scared away by seeing the answers. It's just how you learn. And I hope it was easy for you, somehow. Now, this is the next question which can crack up your mind, and that is question number 43A. Categorize the host cells A, B, and C into transformants, non-transformants, and recombinants and non-recombinants. So that is your task over here. Recombinants and non-recombinants. So over here, if at all you see, I had taught you, I had taught you this particular thing in the recorded format. I told you there is part 1. If at all you are still planning to go ahead with preparatory examination for the next preparatory examination, always remember there are 2 parts of the video that I made. Part 1, 70 on 70, that was basically a live lecture where I covered few chapters, and then there was part 2 which was recorded, and I saw that part 2, not too many people watched this video. For live, so many children were there. I was so happy to see that. But part 2, I was genuinely sad that in recorded, when it comes, you guys do not put that effort to watch the long videos. You need to understand, you get questions from there, and this is the actual proof for that. So what do you call a transformant is something that will take up a plasmid. Okay, which is ready to take up a plasmid. Now this plasmid can contain my gene of interest, cannot contain my gene of interest, that is not the discussion. If at all it is taking up any plasmid, it will be termed as what? A transformant. So over here, if at all you see, there is a vector in this particular thing. That means a vector has come from somewhere. So this is a transformant for sure. What about this one? Host genome and the vector is there. This is also a transformant because again, my dear children, vector is here. What about this cell? This cell just has the host genome. There is no vector. There is nothing extra. So there is no transformation. So this is a non-transformant. These two, my dear children, A and B, they are transformants. Okay, this has not taken up anything, so non-transformant. Now, inside the transformants, there will be recombinants and non-recombinants. You get it? Recombinants, okay. Van Hunger, I'm coming to you directly. Just give me a second, okay? How do you find the recombinants? Recombinants are the one which has the gene of interest. That means the plasmid containing my gene of interest has went through inside. Then it will be, for example, this cell is going to be recombinant. Okay, here the new type of DNA is being entered. Here the DNA is there, but not the new type. It's just the vector DNA which is there. So this is a non-recombinant. The one that does not have my gene of interest is what? A non-recombinant. So I hope you got it. Both of them are transformants. This one is a non-transformant. Over here you have the recombinant. Okay, this is the recombinant A1, and B and C, basically they are non-recombinants. B and also C, it is non-recombinant. It is non-transformant and non-recombinant as well. Got it? Now, define the following terms: transformant host, non-recombinant host, recombinant host. So the same thing, transformant host is a host cell, usually a bacterium, that has successfully taken up any foreign DNA. Okay, any foreign DNA. Now, non-recombinant host, a non-recombinant host is a transformed host that contains only the original vector without the foreign DNA inside. That is without my gene of interest. If at all you have my gene of interest, it will be recombinant host. Okay, we discussed it in this session. This is also a direct question. So for those of you who are saying me that, ma'am, it was kind of difficult over here, you should understand that we are lagging behind somewhere. We are not covering the concepts, and we need to do that, and we have time. Okay, you have time, enough time to do that. Please do that. It is going to be easy. Okay, Now refer to the genetic code dictionary table and answer the below questions. So these are the questions that you have. Someone is giving me so many hearts. Thank you so much. For those of you who are giving me hearts, a big heart back to you also. Okay. So over here, the genetic code dictionary is given. Now you have to give the following answers. Let's see. List the number of amino acids coded by one codon. Only one codon. That means the amino acids which are coded by one codon. For example, if at all you see methionine. Methionine is only coded by AUG. None of the other codons code for methionine. Similarly, tryptophan which is there. Tryptophan which is there, it is only coded by UGG. UGG. So this is there. Okay, this is there. Fine. Dear, can you reload the video? I think that's a problem. I think that's a problem with your net. Can you just check it once more? Okay. Subhash, I hope that's okay. Yeah. Now, going ahead, you have the ones that are coded by two codons. So if at all you see this picture, I can see phenylalanine is coded by two codons. Leucine, can I say it's coded by two codons? No, over here also, these codons also code for leucine, so I cannot take it. So phenylalanine is one such thing. Now you go down the lane. Over here, there is no such things. Coming over here, you have tyrosine. You have tyrosine which are coded by two codons. Do you see tyrosine elsewhere, TYR elsewhere? No. And then you have histidine coded by two codons. You have glutamine coded by two codons. Asparagine coded by two codons. Lysine, two codons. Okay, aspartate, two codons. Glutamate, two codons. So you have to mention that. You have to mention that. Serine is not the one. Serine over here also you see, serine over here also you see. So there are a total of 6 codons out there. What about arginine? Arginine is over here, arginine is over there also, so you cannot take it. What about cysteine? Cysteine you can take. Okay, it's also coded by two codons. Okay. Lost mark here because I couldn't see TRP. It happens. Ma'am, what is your age? Great Thomas says, this question looks big but very, very easy. Exactly. It is very, very easy. Just have to pay attention. Okay. Ma'am, what is your age? Is there an option to skip that question? You can keep guessing my age. It's not a problem. Okay. Shall we go ahead? Now, what do we have? Yeah. The 4 codons one, so you have to mention that. I have mentioned it over here, so you don't need to do much of the things. 3 codons is isoleucine, 4 codons is alanine, proline, threonine, valine, and glycine. Okay, 6 codons is leucine, serine, arginine. Codons with identical bases, that is also a question. Codons with identical bases, that means all the bases are same. For example, U, phenylalanine, A, lysine, C, proline, G, glycine. So these are your things. Okay? Do we have to write the full name of amino acid? No, you did not. You need not do that. Okay, whatever is given over here, you can just mention that. I know it's very difficult to remember all of its name. Okay? Dheeraj, all of the above, dear. All of the above is the correct answer. Okay. We have to full name. Okay. Fine. Let's go ahead then. Yes, Shubhas, please ask me the doubt. Okay. Ayo, question got entered. That's all. Identify and list 4 amino acids coded by degenerate codons. Identify and list 4 amino acids coded by degenerate codons. So basically, degenerate codons, you have to degenerate codons. You have already studied the ones which have multiple codons are degenerate codons, right? Okay, degenerate, yeah. So basically, suppose over here, leucine. Leucine is an amino acid which is coded by 4 codons or basically 6 codons. So this is an example. This is one example. And now, ma'am, they asked to mention only the numbers, whether we should write full name. Okay, just give me a second. Okay, I'll read it. So, list the number of amino acids. Yeah, that is also fine, if at all. Okay, while you number it down, if at all you are putting the terms also, it's totally fine. That's not a problem. That's why I'm saying you can put the number. I didn't see that part. Okay, I actually didn't see that part. So you can just put the number, but for your safety only, while you write down the terms like this, you will make sure that you're not repeating the, recalculating it again and again. So that's for you. You can write it if at all you want, or if you don't write it. It's as simple. Okay? Okay, fine. And Sailani, why are you not listening, ma'am? What, dear? Tell me. Sailani, where are you, dear? What did you ask me? Tell me. Okay. Okay. So yeah, so you have to talk about, so leucine, serine, arginine and valine are the ones that are coded by degenerate codons. All of them will be having multiple codons, that is why they are degenerate codons example. Okay, I hope that is super clear to you guys. So with that we have officially completed, and now I was seeing some question was KVUS smart class. Ma'am, 13th question. So ma'am, 13th question, let me see what is the 13th question. Okay. Okay, this is the 13th question. Is this the question that you were asking, dear? So, which of the following graphs illustrates the population growth curve of a single bacterium growing in a flask of ideal medium at optimal temperature over a period of 24 hours? Rupashri, I wrote in all this correct, ma'am. Oh, that's so, I'm so super proud of you guys. Flask is a limiting feature. But you are growing it only, okay. Just let me check it out. Okay. So KVUS smart, I will just recheck this. Okay, recheck this and I will let you know. So usually since they have not mentioned, so here in this particular question, for example, if you see, this is a similar type of question that was done. So Jiva was growing a bacterial colony in a cultural flask under ideal laboratory condition where the resources sooner or later became limiting. This was being mentioned, which is why it was thought that it is what a sigmoid curve that was going to be followed, or logistic growth that is going to be followed. But over here there is nothing mentioned about limiting features. There is no mention about limiting features out there, and the time is also specified as 24 hours. But since you guys are saying, I'll just recheck it once more and I'll let you know in the comments. Okay, will that be fine? Over here they have not, listen, they have not told you anything about limiting resources, so which is why according to me D1 should be the correct option. But still okay, so A1 shows that it is getting limited after some time, or basically reaching a level after some time. But I believe over here, since there is no limiting option that is given, which is why it should show exponential growth. But still, I will, yeah, 24 hours they kept in that. It can grow as much as it can. There is no limiting thing, right? So according to me, it should be exponential. But yes, I'll still check once, because you know what, in PU2, according to the PU2 board, they have given really different answers somehow. So I will check it once more and I'll let you know. Okay, so according to me it is D, but I'll just check through. If it is not D, then it should be this. Okay, I'll check. Fine. Yes. So with that, I end up this particular session. I hope it was helpful for you guys. I hope you have subscribed to the channel as well, if at all you have not. So my dear children, the next time some examination comes up, definitely do not be worried about any single thing out there. We are here to help you out. Have that trust, and I showed you the way the questions have come. So many questions have been repeated, right? So yeah. Okay. KVU smart classes, I'll Yes, they have given the answer keys as in to the teachers out there. So yes, I'll just check through. Just give me 1 minute. I'll just check. Just give me 1 minute, okay? I'll be right back. Be here. So coming. Okay, dear children, so I am yet to receive the answer for that. Okay, so I'll let you know definitely in the comment section, so please do not be worried. When I get the answer, I'll definitely pass it on to you as well, okay, with an explanation. So Subhash is asking, ma'am, will you please repeat the explanation of question number 44? What is question number 44? Okay, this one. List the number of amino acids coded by one codon. So you have to talk about, so over here you go through the list, okay, and see which amino acid is coded by just one codon. So if at all you see methionine is one such thing, okay, and then if you go here, tryptophan is also one such thing which is coded by only one codon. Fine. Similarly, for 3 codon, how many are there? You have to calculate that. So wherever you see three codons coming to give to code for one amino acid, so you have to give an example for that. Okay, that is a general thing that is there. So yes. Silani, is it fine? And also Subhash, dear, did you get it? And over here I have given you the answers also. Two codons, it is phenylalanine, tyrosine, cysteine, histidine, glutamine, asparagine, lysine, aspartic acid and glutamic acid. Please click that, that's all. Ma'am, I wrote 39 like chart of HIV. 39, 39 is what? Chart of HIV. Yes, yes, yes. The schematic representation of HIV, if at all you're reading it, then it's okay. Okay? Rate Stormer, ma'am, big heart for reading all our chats in your live. Thank you. You're so sweet. Thank you so much. Ma'am, also that dihybrid MCQ, the answer is capital R, small r, capital Y, small y. That is the C option. That is for sure. That is from PU2 board. Confirmed. Okay. Ma'am, they should be 2, 12, 1, 8. 2, 12, 1, 8. Subhash. 2, 12, 1, 8. Let me check. So over here, if I see 2, okay, codons, 1 codon, 2 codon, 3 codon, 4 codon, 6 codon, and the codons with identical base. So 1 codon is 2, and here for 2 codons it is 9, for 3 codon it is 1, for 4 codon it is 5, and for 6 codon it is 3 amino acids. Okay, just check the name with this list, and you will understand it better. Ma'am, 4th question answer, okay. Okay, should be the answer. Subhash says, ma'am, all of this should be the answer for the dihybrid. It is not. Okay, so general terms, even as teachers, we also got confused. I went ahead and I asked other teachers also, and then they gave me the thing that yes, the PU2 board said the answer for the question is C, capital R, small r, capital Y, small y. The reason being that that is a true dihybrid. That is a true dihybrid where there are 2 features out there. Okay, just a second. I'll come to that question and I'll explain you this. So when I went ahead, I went with all of this, but it is not for the same reason. Why? Because if at all you do not have this dihybrid, then you will not get a cross like this where 9:3:3:1 ratio will be coming in. So to validate this particular ratio, we need to have parents like that. So we consider this to be a pure dihybrid out there. So I definitely get it. I also went with this, but yes, according to your PU2 board paper, this is the answer. Okay? Fine. Ma'am, 6th one. 6th one is saltation. It's a very easy question, right? Very, very easy question. Cool. Now, the only question that you have a doubt in, I will just confirm you and I'll let you know. I think rest of all the questions have been covered. So PU board passes the answers to the respective school authorities, as in the teachers. For this, answer keys will be there. They have already got it. So yes. Last question of 5 mark. You want me to explain it once more? No, Rupashri, they have given, I could not see it. That's the only thing. I could not see the answer. So I asked another teacher for the other questions answer, which is how I got it. Okay, I asked them because I had a doubt. I went there and asked them. That's how. But for this question, I didn't have the doubt. I thought that D will be obviously the correct answer. But since now you have come up with a lot of things, so I also have a doubt now. I have passed the question on. Okay, so once I get the answer, I'll get it back to you as well. Yeah? This paper was easy, made us difficult. Okay, so yes, I think that's all. Ending up, my dear children, with the life. I hope it helped you in some or the other ways. And let's come back, let's meet in the second preparatory exam, if at all it's there for you guys, or your direct board examination. Ma'am, 11th, oh my god, guys, go scroll back. Okay, 11th is what? 11th answer is stirrer. According to your NCERT, it would be stirrer. Just a second, okay? Fine. So yes, it's not impeller, it is stiller. I thought I got the answer. So I've got one confirmation. The answer for that would be exponential, but still I'll be checking out. So the teacher whom I asked said it is exponential, but still I'll be checking and giving you the answer. Okay? 8. What is 8? 8 is this: injection of antitoxin in tetanus confers which type of immunization? You're giving ready-made antibodies, and that too artificially you have been getting it. It's not what do you call? So your answer for this is artificial passive immunization, not natural. Natural is when you get from your mother to the child and things like that. Okay, namaste dear. Fine. So fourth question answer is this. I already told you. So, okay, so I'm ending up the live with a perception that for this particular question, you might have a doubt between sigmoid as well as basically exponential curve. I'll just confirm and I'll let you know. Okay? Fine. So, yes, flask can be a limiting factor. So, yes, I'll let you know about that in the comment section. This is all for today, my dear children. Wishing you all the best. And yes, last question, offline section 1 mark, didn't understand. Okay. Thank you, Subhash. Okay. Please let it be D only. I'll check through. Okay. See my question. Okay. Wait. Are you asking about this question? Who are you? First of all, Sailini Sheikh. You're asking about this question. I explained it a plenty of times. I can explain it once more. Okay, I can explain it once more. Listen to me very carefully. Here you asked, list the number of amino acids coded by one codon. Okay, one codon. Only one codon should be there. Methionine, only one codon. AUG, do you see methionine elsewhere? No. Similarly, tryptophan, only one codon. You do not see anything elsewhere. Now, the ones which are coded by two codons. Histidine, glutamine, two codons, tyrosine, 2 codon, asparagine, 2 codons, lysine, 2 codons, aspartate, 2 codons, glutamate, 2 codons. Okay, and over here, if at all you move phenylalanine, 2 codons right up over here. So these are the examples for 2 codons. Similarly, you have to go with 3 codon examples also. Okay, 3 codons. Okay. Section 1. Yeah. Section 1, 5 marker starts from here. Which question? Ma'am, I got 6 MCQs wrong. Ayyo. What are degenerate codons? Degenerate codons means the amino acids which are coded by multiple codons. For example, UUU code for phenylalanine, and UU, what do you call, this one also codes for phenylalanine. So these are degenerate codons. The codons which are coding for what do you call, amino acids. Okay, multiple codons are coding for a single amino acid. Okay. Okay. Fourth question, discuss, ma'am. Baba, I had already discussed it. So, actually, in fourth question, this should be your answer. Where is it? This should have been your answer, but this is the only correct answer. C is only the correct answer. Why? Which of the given genotypes are dihybrid over here? Only this, because this one proves the validity of 9:3:3:1 ratio. If at all you have any of the other parents, it won't give you this ratio. That is why exclusively PU2 board considers this to be the correct answer. Even I do not exactly go with this thing, but still, when I went with all of this, but yeah, according to the PU2 board, whenever you have two of them has heterozygous, okay, capital R, small r, this is heterozygote, this and this is also heterozygote, you will call this as a true dihybrid. Okay, fine. Chalo. So I am ending up this particular live right over here. Now I am not going any further. And also one more thing. Yes, that's all. So my dear children, wishing you all the best. Thank you so much. Please take care of yourselves. I hope this had helped you. Okay? Fine? In F2 generation, all of them come, but who is the parent? It is capital R, capital Y, small y. That is the dihybrid that you have initially got, and that gave rise to all of it, right? So that is the thing. I agree to your point. It's not so smoothly correct. So yeah. Practical exam, we will come with so many things for practical exam as well, Vittal. So don't be worried, okay? My lamb, thank you so much. Thank you so much, Alina. Everyone, thank you so much, dear. And yeah. Wishing you all the best. Bye-bye.